First of all, let me say thank you for inviting me here today. I'm going to talk to you about honey. It's something that I've been working on since the 1990s. We've done an awful lot of work in the laboratory, and what I want to do is show you some of the work that we've been doing and perhaps explain to its relevance to clinical practice, but to take a broader view and basically to try and evaluate what evidence there is for the use of honey in wound care, and perhaps to see what some of the limitations are. So I'm going to actually have a look what could be claimed for honey. How could it support wound healing? What kind of evidence is there for that? And I'm going to look at it particularly from a laboratory point of view, and then to see how that translates into clinical practice, and then to look at some of the weaknesses in the evidence. So, honey is basically, as I'm sure you all know, an ancient wound remedy. It's been used for thousands of years, and there are some communities that never stop using it if you look at it on a global scale. In the UK, it was certainly used up until the 1970s in British hospitals. I know that there are some people here today who will have trained and used honey from jars on hospital wards. Now that kind of stopped when we had the development of modern wound dressings, and we kind of set honey aside, forgot about it. And now we have increasing difficulty treating infections because of antibiotic resistance, and we have to perhaps look at some alternative remedies. So we're kind of going back and looking at some of those ancient remedies to see were they any good, and could they be used in the modern situation. So there was a lot of work being done in New Zealand and also in Australia in the 1990s, and I became involved with working with honey in the 1990s. And the first product that was actually developed, this one down here, Medihoney by Anthony Maloney in Queensland, was a blend of manuka honey and an Australian honey. That was licensed by the Therapeutic Goods Administration in Australia. And it's for the first of what I call the modern products. We had a product developed in the Netherlands that was based on a World War I remedy, a German remedy, and that was licensed in 2003. Honey was introduced into the UK in 2004, and now it really has begun to take off. It's throughout the developed world, you begin to see countries using honey for the first time. In 2007, Canada and FDA in America gave approval. And I'm sure you're going to hear, actually, in coming years, a lot more about honey. And so people then started to review what is the evidence. And so I'm going to talk about some of the evidence and the Cochrane reviews a little bit later. Now, there are actually quite a lot of honeys that are being used in modern practice, in the licensed products. Some companies tell you what kind of honey, floral honey it is. Revamil, which is produced in the Netherlands by a company called Bee Factory, actually have got bees under polythene, and they've got herbal crops there. They don't tell you what they are, but they produce a very consistent product because it's produced in a very consistent way. A lot of products contain manuka honey, which basically comes from New Zealand, although there are varieties that similar honeys that are being generated in Australia. A few years ago I was in Slovenia. They've chosen to use chestnut honey. Buckwheat is produced in Canada and the States in huge quantities. And more recently, France, a company in France has developed a product using thyme honey. So there are a wide variety of honeys that are being used. And in the last 10 to 15 years, certainly since the turn of the century, this term has been developed called medical grade honeys. So these are the sorts of honeys that are being used in modern products. And basically, they are ones which have been demonstrated to have antibacterial activity using laboratory tests. They generally are produced under hygienic conditions to minimize contamination of the honey once it's harvested. All of the honey is traceable, because when you've got a medical product with a code on it, you have to be able to actually trace the origin of all of those products in case there is a problem. And so that is certainly the case now with the products I'm talking about. Generally, they tend to come from fairly remote areas, which will obviously reduce the extent of any contaminants, pollutants, certainly things like pesticides. And we don't really want any antibiotics in our honeys either. For eating honey, there was a bit of a scandal in the UK some years ago when honey from China was, for eating purposes, was found to contain chloramphenicol. You may remember that. So now all honeys are screened for any traces of antibiotics. And of course, we're talking about a modern product now, so that's irradiated by gamma irradiation, because naturally honeys do contain some microorganisms. So if you were to plate out medical grade honey in laboratory media, nothing should grow. But if you took honeys from the supermarket and plated them out, you certainly would find microorganisms. Now, by and large, a lot of those are non-pathogenic, but actually we have been able to find occasionally potential pathogens in honey. So you don't want to take honey off a supermarket shelf and treat your own leg, although I know a number of people do. Not nurses, patients. Much of my work has been influenced by Peter Molan from New Zealand, who's written a great deal about honey and I think first brought our attention to manuka honey. And basically, the therapeutic claims for honey could be divided into two categories. A, its antimicrobial properties. So its role in perhaps treating infection or preventing infection. And one other important thing there is the fact that it can deodorize it. It actually removes odor, which is a sign of infection. But the second category, and I'll talk a little bit less about that because I've mainly concentrated on the first one in my own work, is that it does seem to promote wound healing. You hear a lot of people say that when they first use honey on a chronic wound, and often honeys are used on chronic wounds, not exclusively, but it kickstarts the healing process. So could it indeed have properties that can interfere with that process, influence that process, perhaps would be a better way of putting it. Now, Peter Molan wrote about that in the Journal of Wound Care back in 1999. And so what I'm going to do is look at what is the evidence to support those claims. So let's have a look at the antibacterial properties first of all. Well, honey is what we call a broad-spectrum antimicrobial agent. It's got a really high sugar content and very little water, and even the water molecules in sugar, in honey, is locked up with the sugar molecules. It's not available for microbial growth. Also, honey is fairly acidic. So just the high sugar content, low water content, and acidity in honey means that all honeys have got antibacterial activity. You probably know you can keep them in your larder for years and they don't tend to go off. I mean, jams get mouldy, but very rarely does honey do that. But there's more to honey than that, and it really is. Chemically, it's a quite complicated substance. Way back in 1919, people realised if you dilute honey, it actually gets more potent. It took some time, but some group of workers in Cornell University in the 60s showed that that's because when you dilute honeys, there's an enzyme there that oxidizes glucose and generates hydrogen peroxide at very low levels. Now that is an antiseptic, and we're talking about very low levels, about 1,000 times less than when we used to put hydrogen peroxide solutions onto wounds. In 2008, methyl glyoxal was discovered in manuka honey. It seems to be at very elevated levels in manuka honey compared to other honeys. And some work in New Zealand has shown that the precursor for methyl glyoxal is found in the nectar of manuka bushes. So if you take the nectar, you can find the precursor. So that would explain why there are high levels of methyl glyoxal in manuka honey, and that too has some antiseptic, antimicrobial properties. Then the people working with Revamil honey and found some antimicrobial peptides called B-defensin. In Canada, Katrina Brzezinski's been working on another kind of antimicrobial that is generated by complexes formed between proteins and sugars, and she's called those melanoidins. Recently in Japan, a glycoside's been discovered in manuka honey called lepticin. I think we're going to go on and on and on finding more and more bioactive components in honey. It's a really complicated story. The other thing is that all honeys have got antioxidants in them, and antioxidants themselves have got antimicrobial activity at low levels. So there's this really complicated story that there are lots of antimicrobial components in honey in general. But the trouble is, not all honeys are alike. Now, we were asked to do a study by an American company just looking at supermarket honeys, because when people started to hear about the use of honey for wounds, and we've had an awful lot of publicity, because when I started this work in the 1990s, it was all very wacky, and the journalists thought it was a good story, it would interest the public. So if we've got a new antimicrobial agent under development, the public doesn't get access to that until it's licensed. But once they hear about honey, they kind of might have got the idea that they could treat themselves and go and buy their own honey. So we took some honeys out of the supermarket, did some tests in the laboratory to see if they all had the same potency, and of course they didn't. 10 of them didn't even have any detectable activity in the tests we were doing. We found that some manuka honeys that were claiming to have higher potency than they actually had, according to the label, and of course those honeys weren't sterile, and we did find quite a variety of organisms in them, some potentially fatal. And we had a medical grade honey in the study, and that actually had the highest level of potency. Same sort of study has been done in the Netherlands by Paulus Kwakman, and he's compared manuka honey with that honey that I talked about that comes from polythene greenhouses, and showed that they do have different activity. Basically, one has got high levels of hydrogen peroxide, that's the Revamil source honey, but manuka honey doesn't seem to have high levels of hydrogen peroxide generation. The Revamil honey has got B-defensin, where we don't seem to see high levels of that in manuka. Low levels of methyl glyoxal in the Revamil source, and high levels in manuka. So not all honeys are alike. I mean, you've only got to look at them in the supermarket. Some are dark, some are light, some are runny, some are set. They come from different plants, and there will be different phytochemicals in those honeys. Also, recently a study by Chen has shown that chemically honeys change with storage. So the longer you keep them, the more they change. And the way you keep them too will influence what happens to them. If you leave them sitting in the sunlight, in the light, they're more likely to change than if they're kept in the dark and a cool place. So basically, it's really quite a complicated story. Much of my work has been done with manuka honey, and we've been fortunate either to get it from Peter Molan in New Zealand, or from companies that use it in their dressings, and they've given us sterile honey, which obviously helps us in our experiments. So we've basically been looking at does it kill microorganisms that come from wounds, and I've got a large collection of bacteria that have been isolated from wounds. The answer is yes, it kills Staph aureus at very low concentrations. And when we started this work, everyone said, oh, well, of course, it's the sugars in honey. So it's not just the sugars in honey, it's those other components that contribute. It kills MRSA, enterococci, coagulation-negative staph, and beta hemolytic streps. So basically, antibiotic sensitive and antibiotic resistant organisms got the same sensitivities, and other people have done similar sorts of work to confirm what we're saying. The same with gram-negatives. We need a slightly higher concentration to kill them in the laboratory, but again, a lot of work's been done on Pseudomonas and other enteric bacteria. Now, you don't have to dilute your honey to use it. You're going to use it either neat, or the way it comes in a dressing, it's going to be at much higher levels than we need in the laboratory to kill the bacteria, which is fine. How does it inhibit the bacteria? Well, my students have done an awful lot of work here. My first PhD student was Anna Henriques, and she showed that if you expose Staph aureus to honey, you get these organisms that accumulate with this cross wall. And you see here, this is an electron micrograph, and these are bacteria that have been exposed to honey, and basically they can't divide. Now, this is work that Rowena Jenkins did after Anna had finished. This bacterium is a round bacterium. It grows within half an hour. It then divides. It lays down a membrane across the middle of the cell, then cell wall, then some enzymes come along, digest the cell wall, and the two daughter cells separate. So basically what we were seeing is organisms that were locked into that position there. They were stuck at that position. And what Rowena was able to show is those autolysin enzymes that normally break down the cell wall are inactive. So they don't function. So basically, if this organism can't grow and divide, it can't go on to form an infection. And here's some of the pictures that she produced. The top are either controls, that's no honey, that's artificial honey, that's 2.5% honey, so this is below the inhibitory level, and then we've got 5, 10 and 20% honey. And you see, these are inhibitory concentrations. You see the cells are bigger and they are locked in, they can't divide. And we did one last control with methyl glyoxyl, so it's not methyl glyoxyl alone, it's probably methyl glyoxyl working with something else that inhibits them. In Australia, they've looked at the proteins to try and work out what was happening inside the cells, and this agent has a unique mode of action. We recently did quite a complicated study where we looked at the genes that were being expressed with and without honey. And the important thing is that this last point of the slide that I want to emphasize is what we found is the genes that control virulence and biofilm formation are knocked out by manuka honey in MRSA. So even if this organism is present, it can't express its virulence determinant, so it's unlikely to cause an infection. The first reports, does this relate to clinical practice? Well, Keith Harding in his clinic many years ago, 1999, did treat a patient and cleared MRSA from that patient. Similarly, Gerald Dunford did the same thing in Salisbury, and I'm happy to report that throughout the world other people are beginning to report the same thing, eradication of MRSA from various different types, mostly chronic wounds, but not always. Certainly some acute wounds too. And this, I'll run through these very quickly. This was, again, one of our first cases that Gerald Dunford treated. This patient had had meningitis, and these lesions had not healed for 9 months. They had multiple infections there. There was a Pseudomonas, a staph, and an enterococcus. And in 8 weeks, this guy healed. But importantly, odour was controlled. Same sort of thing, can we inhibit gram-negatives? Yes. This shows that the bacteria are very shortened when in the presence of honey. Also, the cell wall is affected. and Aled Roberts, my last PhD student in this area, has shown with this dye, live cells without honey are green, and when you treat them, this is only for 2 hours, you see there are very few green cells left, and the red is dead cells and the contents of the cells. So these cells are bursting open, and so you've got a different mode of action for different bacteria with honey, and he's shown that it's due to changes in the cell wall, and he looked at those genes. Can it interfere with biofilms? So many people are interested in biofilms. There are 3 potential interference points in the biofilm life cycle, and certainly there are now studies that show that honey can prevent bacteria sticking to a surface. They don't stick, they won't go on to form a biofilm. Also that it does interfere with quorum sensing in gram-negatives. Some of that work was done in Spain and some in Harvard. So again, that will control virulence, and it will control the ability of a biofilm to develop into a mature biofilm. And finally, does it disrupt a biofilm? Well, yeah, there are a few studies throughout the world, but I just want to concentrate on one that we've just recently published. This is a biofilm with no honey, 20% honey. You see the cells are increasingly shortened, 30% honey, 40% you actually see disruption of the biofilm, and at 50% honey there are virtually no live normal looking cells left. Again with the red-green dye, no honey, lots of green cells are alive, 20% honey, you've got disruption already, 30% lots of dead cells, 40% very few cells left at all, and at 50% most have gone. Still 1 or 2 live cells. Those are probably what we call persisters. So that's been confirmed in Staph aureus, again by the group in Sydney. So a quick look, what about these therapeutic claims? Is there any evidence to show that honey could modulate those cells that are important in the wound healing process? Well, yes, there are increasing studies. We did some work back 10 years ago. Some of our PhD students put honey onto monocytes in the laboratory, and we could show that the suppression of reactive oxygen species was depressed, and also that you could get synthesis of pro-inflammatory cytokines. So this shows that honey is immunomodulatory. And if anyone wants to read more about this, there is now a review that's just was published last year by Uri Makian down here. In France, they've looked at the stimulatory activity of thyme honey on macrophage using a mouse model, and in Slovakia they've shown that keratinocytes cultured in the laboratory are stimulated to produce cytokines under the influence of honey. So there is increasing evidence. We already had shown that it suppresses the production of reactive oxygen species. And finally, Peter Molen has shown that it has a role to play in debridement. Now, when you've got fibrin deposition in a wound, which in fact you do tend to get in a chronic wound, chronic wounds actually, macrophage in these wounds produce an inhibitor, because in order to get fibrin deposition, it's basically achieved by plasmin, and plasmin is produced as a precursor. So you have to convert that into an active enzyme to break down fibrin. Now, macrophage in a chronic wound produce an inhibitor that actually removes that activator. So therefore, the inactive precursor is not converted into the active enzyme, and so therefore you get build-up of fibrin. It's not removed as naturally. In the presence of honey, that inhibitor is removed, so therefore we don't get removal of the activator, so that would promote debridement. So we're beginning to get mechanisms that explain those therapeutic claims. But what I want to do in the last part of my talk, and I've only got about 10 minutes left because I don't want to encroach on your coffee time, is to relate that to clinical practice and what we call our pyramid of evidence that supports evidence-based practice. We've got various levels of evidence, and you see in my pyramid, laboratory evidence doesn't even feature. I would have thought as a scientist, knowing how something works would influence your choice of an agent. Also, you'll see that a single case study isn't there at all. You'd have to have a case series. The best kind of evidence, of course, is an RCT. But the thing is, once people start to do RCTs, they maybe several accumulate, and then, of course, they're done in slightly different ways. So Cochrane, Professor Cochrane decided to use a device, a systematic analysis using, so that you could sort of evaluate the clinical evidence. So the best kind of evidence comes from systematic reviews and meta-analysis. So what about those claims? Actually, there's not an awful lot of evidence to support the role of honey in clearing infection. I've got in vitro data, doesn't come on the pyramid. Case study isn't important, and although there is a lot of anecdotal evidence, the same when it comes to does it promote healing? Not an awful lot of evidence, but there are some RCTs. So let's have a look at how they've been reviewed. There are three main reviews, although some people have actually, Professor Molan has written a great deal about clinical evidence. The first one was done in 2000 in Oxford, and then we've had 2 Cochrane reviews. Now, when they did the first review in 2001, they included only 7 RCTs. I think actually there were more available, but they just weren't noted. And one of the criticisms was that of those 7, 6 were done in India by the same person. So there is a bias in that maybe somebody is in favour of honey, and kind of it kind of negates to a certain extent the evidence that they're producing. But basically the analysis from this review showed that although some of the outcomes were superior, there really wasn't firm evidence to support the use of honey in wounds, although the authors did say that there was some biological plausibility. The first Cochrane review, which was published in 2008, included 19 RCTs. So we got acute and chronic wounds. And because there were so many studies involved and so many different methodologies, they weren't able to do a meta-analysis, which is a kind of a way of producing a figure, a number to evaluate those studies. And they basically said that the quality of those trials was relatively poor. And I'm not criticising that observation. I think many would agree. Their conclusions were that it may improve healing time in some cases, particularly with burns. Essentially, they said there was not enough clinical evidence to guide clinical practice. The same kind of exercise was done more recently by Broleman and colleagues. And what they did is they looked at the broad range of evidence relating to the clinical use of antimicrobial inventions in wounds. They screened 149 systematic reviews, and they took 44 of those to look at them. And they actually came up with a whole series of conclusions. And although they've been looking at the same evidence as Dual, came to a different conclusion and said that there's strong evidence for 16 interventions, and one of them was the use of honey in burns. So that here you have 2 people looking at the same evidence but coming to different conclusions. Now, the whole point about a systematic review is that it should be objective and it should be transparent. Ideally, everyone ought to come to the same conclusion. the Cochrane review for honey was repeated, updated in 2003. Now, this is only 5 years since the first one, and in actual fact, there was only an additional 6 RCTs, and the number of patients only went up by less than 500. But they actually came out with quite strong conclusions in this report. Basically, they said that honey dressings don't increase the rates of healing, and they even said that they may delay in partial or full thickness burns. Now, those burn studies, all but one of them, had been included in the previous systematic review by Jewell, and they didn't actually make such strong conclusions at that point. What they did do is they included one particular study on leishmaniasis, which is a protozoal infection in tropical areas where you get basic insect bites and then a chronic wound develops. So they included that one. Some people must say that that's not really considered to be a typical wound. I think the most important thing was that they did say they were superior to some conventional dressings, but again, there's uncertainty, there's not broad evidence, and I think actually most of the Cochrane reviews come out with those kinds of conclusions. The trouble with the Cochrane review is the primary outcome that they're mainly concerned with is healing. Does the wound heal? And obviously there are other things that might be included. But I think the thing that shocked me most about this last review was this last statement here, that there was insufficient evidence and they are actually advising purchasers to refrain from providing honey dressings. Now, I've never seen a Cochrane review actually make a recommendation like that before. It's meant to be provide the evidence and let you make the decision. So this has been criticised. I wrote some comments that were published recently about that last review. To my mind, what they're doing is they've included a lot of studies that were performed before we had medical grade honey. They actually include a wide range of honeys. They came from places like Egypt, Iran, India, Malaysia, Pakistan, Yemen, and of course the UK and New Zealand. So we've got a mixture there of, shall I say, local products and medical grade products. They came obviously different kinds of honeys. They would have come from different floral sources. And when you begin to analyse the chemical compositions of honey, you see perhaps that maybe they're comparing apples and oranges. Some of the RCTs actually do have unusual comparators. There's one in India that was done with burns where they compare honey with boiled potato skins. That's of no relevance in the UK. It may be relevant in India, but not in the UK. I think it's actually, and they also did criticise the blinding procedures. Now, to my mind, it's always going to be really difficult to have a double-blind RCT when you've got honey. You can smell it, if nothing else, so the patient knows that they're being treated, and probably when the clinician does when they're doing the assessment, because sometimes when you take the dressings off, there's still some yellowness on the wound. I mean, could we ever have a placebo that would mimic honey? I don't know. And also, in the last, in 2003, Jewell actually criticised the randomisation that had been done in the burn studies in India. There was a method called the CHIP method, which he said wasn't suitable. And he actually contacted the author of those reviews, asked did they use envelopes and in the randomisation process, and Professor Subramaniam said yes. But Jewell actually criticised them because he said they didn't say whether they were opaque envelopes. I've never seen transparent envelopes, so I thought that was unfair to make that criticism at that point. And the other thing is they actually excluded one study in particular because it was used on fungating wounds. They're never going to heal. Okay. But honey does have a role to play for those patients because it's so important in deodorization. So to me, I felt that they were using the evidence in a selective way. Certainly in the States, Janice Beats and Laura Bolton were also very critical of the dual Cochrane review. They went as far as to say that they thought the statements in the conclusions or the abstract were not clearly derived from the results reported. And actually these are their slides. What they said is of the 25 RCTs, 11 did favour honey with statistical significance. And there are actually only 3 that were against, and one of those was the one on leishmaniasis. They felt that most evidence did support the use of honey in dealing with chronic wounds. And actually they did an analysis of the healing times rates with honey and the controls. And if you take out the leishmaniasis study, you get a much better a balanced view. So they felt that the evidence on burns should be qualified. One point they made was that certainly in 1999, one study on burns was done with honey and tangential excision. If you've got a full thickness burns, they compared excision with honey, and honey didn't fare well. But the point that Beats and Bolton have made since is that since 2006, excision has been the standard of care. So to compare honey with a standard of care maybe was not, is no longer appropriate. And what they suggest is if you wanted to really look to see if honey does have a role to play in full thickness burns, maybe a better study to do now would be to have the standard of care with and without honey. Does that make sense? Okay. Actually, Jewel has just this year revised and produced issue 2, and they've taken out that study, that point about advising purchasers not to provide honey. So they obviously have received criticism and they've modified their conclusions. If anyone wants to read a little bit more about this complexity, I've actually just recently published a review on this in a new journal, which is a Chronic Wound Care and Management Research. It's an electronic journal, so you'll be able to find that. And I talk there about the complexities of honey and the difficulty of ensuring its chemical consistency. So there are some advantages of honey. One certainly is that there's no reports of cytotoxicity, but I do accept that the clinical evidence to support its use that's come from clinical trials, particularly RCTs at the present time, is weak. So finally, I'll just say to you that this is a broad spectrum antimicrobial agent. We know from laboratory tests that it has the potential to inhibit organisms at low concentrations, even though it's going to be used at much, much higher concentration clinically. But the important thing is we now know that not only does it inhibit organisms, it actually, if it ever got diluted to the point where it wouldn't kill them, it certainly would reduce their virulence, and that would be important. It prevents biofilm formation in the laboratory. We need now to study that clinically. We need to have wounds which are known to have biofilms that are then treated and then observed later. And at the moment, we don't have a good test for finding those wounds that have got biofilms. So we've got laboratory data, but that needs to be tested clinically. And we need to improve the design of trials so that in future we're going to get some better evidence. And I believe that we should perhaps, when we do systematic reviews in the future, separate out the traditional products from those that have used the licensed modern medical grade honey. Both are still valid because they're still being used in different parts of the world, but maybe the evidence shouldn't be muddled up. Thank you for your attention.